All right, welcome back to our overview of Chapter 7, Membrane Structure and Function. So here we're going to move into Concept 7.4. So active transport uses energy to move solutes against their concentration gradient. Okay, so if facilitated diffusion is still passive because it's moving with its own concentration gradient, and this requires no energy. There are transport proteins, however, that can move solutes against their concentration gradient. Okay, this obviously is going to require some input of energy to achieve because you're causing something to happen that shouldn't happen otherwise. Okay, so this is something that we refer to as active transport, right? So we're moving things against their concentration gradient. And typically this requires some input of energy. Oftentimes it is in the form of ATP. So we hydrolyze ATP, pop off one of those phosphates, use that energy to drive another process. Okay. This active transport is performed by specific proteins. Okay, there are specific proteins that transport a specific item with a specific mechanism. Now, active transport allows cells to maintain particular concentration gradients that differ from their surroundings. So think about the amount of sugar that might be in a cell. That cell needs that sugar for fuel. It's always going to be low concentrations of sugar outside the cell, but it needs to maintain high inside the cell. So the cell is constantly working to absorb sugar against this concentration gradient. Now, let's take a look at one specific type of active transport mechanism. This is something called the sodium-potassium pump. Now, how the sodium-potassium pump works is you take a cell, and here's the inside of the cell, here's the outside of the cell. Outside of the cell, you have large amounts of sodium, but low amounts of potassium. Inside the cell, we have large amounts of potassium, but low amounts of sodium. Okay, and this is mostly achieved by this pump, right? So here what we have is a carrier protein open, and it has very high high affinity binding sites for sodium ions. That is going to mean that it's very likely that sodium ions are going to bind these sites. When that happens, you cause a shift in the protein structure. That protein structure is going to stimulate the phosphorylation of ATP. So we're hydrolyzing ATP, but instead of just releasing that phosphate out, we're actually transferring that phosphate onto the protein itself. Okay, so now we've harvested part of the energy of this hydrolysis, but not used all of it by popping it onto the protein itself. When that happens, that actually causes a conformational shift in the protein. That causes it to close on this side, open on the other, but it also changes the shape of the sodium binding sites. That makes it a low affinity binding site. It essentially pops them out. Okay. At the same time that that happens, we've changed shape of these other binding sites, which are actually for potassium. These are now high affinity potassium binding sites. So potassium will come in and bind because there's something that is holding it in place. So potassium ions will just be bouncing around out here, and sooner or later one will bounce in and fit into the spot. When this happens, that causes this phosphate ion to be released, which is harvesting even more energy. That release actually causes a conformational shift, which reopens the protein to the other side and also changes the affinity for potassium, turning it into low affinity binding sites, which is going to cause the potassium to be released. So utilizing this mechanism, we can continually pump sodium out and potassium in, regardless of what the internal and external concentrations are. So even when we are continually putting more and more sodium outside and more potassium inside, we can still keep driving that process. When this happens, this protein can then repeat its cycle. All right, so this gives us a distinction of passive transport, where we have things just moving through by its own energy. This can be done either by diffusion or through some type of facilitated diffusion. Again, this all utilizes its own energy of diffusion to allow it to come through. That is converse to active transport, where we utilize energy and burn that energy in order to pump things against their concentration gradient. Now, something to consider is when we have a situation where we are pumping ions, there are actually two things happening, right? So when we pump an ion across, we are both moving the ion itself through, which is making a higher concentration on one side versus the other, but it's also changing the overall voltage of the inside and outside of the cell. So here, because we are moving all these positive protons out, we're going to have a net negative charge on the inside and a net positive charge on the outside. You can actually utilize this as a combination of two different forces, right? So we're using energy to move these protons out, and it's creating two separate gradients. One is a chemical gradient based upon the concentration of ions that are going to be outside versus inside, but also an electrical force, right? This is essentially what a battery is. So our cells actively produce their own battery-like structure by pumping ions out. What we can do is by constantly moving ions out and creating both this chemical force, this chemical gradient, and an electrical gradient, we can harness that to do other work. So here we have what's called a cotransporter. A cotransporter is an active transport mechanism, but it's utilizing something else to facilitate movement. So here, this sucrose proton cotransporter can transport a sucrose molecule, which is going to be actually in a lower concentration outside the cell than inside the cell. So it's moving it against its concentration gradient by moving a proton through with its proton gradient. And it only has a high proton gradient and electrical gradient because we've used energy to pump those protons out. So you can imagine that the cell can, and this is particularly the case with plant cells, they tend to use proton ions. We tend to use the sodium-potassium pump to achieve a similar type of effect. But this plant cell is pumping out protons, and then it can have a whole series of different cotransporters that it can utilize the same stored energy, which is this proton gradient, to drive the absorption of other things. So active transport uses energy to move solutes against their concentration gradient. So here we have passive transport. Things can either diffuse through or be facilitatedly diffused through by proteins. Active transport is the opposite of that. We're using energy to move things through. And this can be done either directly through things like ATP hydrolysis to drive a particular protein conformation change, or we can utilize that ATP hydrolysis to pump protons and then use that stored proton gradient, which is a combined electrical and chemical gradient, to drive the import of other things. Now, sometimes we need to move more material than just a single molecule at a time. So concept 7.5, bulk transport across the plasma membrane, occurs by these processes called exocytosis and endocytosis. So recognize that these small molecules in water can be actively pumped or moved and transported by these proteins. We start getting bigger stuff, large biomolecules such as polysaccharides, bigger proteins. This requires more than a channel to fit through. You can't build a channel big enough without losing all your selective permeability. So here we have a bulk transport mechanism, and this is of course going to require energy. Now, letting things out is through this process called exocytosis, and we've actually seen this, right? So this is when we have these transport vesicles that are blebbing off from our various organelles. They move from the ER to the Golgi apparatus, and they can ultimately fuse with the external membrane and release their contents outside. Whenever we have secretory cells, this is typically what happens. So when we're producing mucus or when we're producing milk, anything that we are secreting out oftentimes utilizes this bulk transport process. The reverse of this, endocytosis, is when we essentially do this in reverse. Instead of fusing a vesicle to the outside membrane, what we do is form a vesicle from the plasma membrane and pull stuff in. This falls into one of three categories: phagocytosis, which is known as cellular eating; pintocytosis, which we can consider cellular drinking; and this third process, which is related to pintocytosis, is called receptor-mediated endocytosis. So, phagocytosis, we oftentimes represent as we can take pieces of the membrane, we might bind something on the outside, and then utilizing things such as actin, we can polymerize actin and kind of wrap around this food particle and then seal it off into this food vacuole. We could then take this food vacuole and fuse it with a lysosome to start digesting that material. Pintocytosis is essentially the same process, but we're not actively selecting something or bringing in a chunk. All we're doing is pulling in a little invaginated pocket, and whatever comes sucked in through it is what you get, right? So this is completely non-specific. This occurs through putting a protein coat on the outside, which has a natural geometry of a sphere that's going to force an invagination and the formation of this vesicle. A very similar process happens in receptor-mediated endocytosis, but here the formation of this protein coat causing vessel formation is actually facilitated by some type of ligand binding some type of receptor in here. So this receptor is binding a ligand that is triggering the coat to form, which is bringing that specific item in. You also bring in some of random things as well, but you are bringing in something specific. So here we have concept 7.5, bulk transport across the plasma membrane occurs by exocytosis and endocytosis. This involves either the fusion of vesicles with the outside membrane releasing stuff, or the formation of vesicles from the plasma membrane pulling stuff in. And this occurs through phagocytosis, pinocytosis, or receptor-mediated endocytosis.